Hi everyone, I'm Susan Limbu from University of Manchester and I'm one of the authors of the paper titled A Folliculocentric Perspective of Dandruff Pathogenesis. Could a troublesome condition be caused by changes to a natural secretory mechanism? The article provides an up-to-date understanding of dandruff pathobiology with a folliculocentric perspective, and we introduce a novel working hypothesis to dandruff pathogenesis. Dandruff affects more than half of the world's adult population, and the symptoms, which includes flaking and itchiness, is not only an annoyance to the sufferer, but it can also affect their self-esteem. As dandruff occurs on scalp only, the scalp consists of skin and hair follicles. Normally, the scalp consists of commensal microorganisms such as the yeast, Malassezia species, and bacterial species such as Staphylococcus epidermidis and Cutibacterium acnes. Mites such as Dermodex follicularum are also present on the hair follicle. The hair follicle has its own immune microenvironment. There are several different types of immune cells, such as Langerhans cells, CD4 T cells, CD8 T cells, and mast cells near the infobidulum, which starts at the opening of the hair follicle to the start of the sebaceous gland. Along the hair follicle, there are mast cells, macrophages, and few CD4 T cells and CD8 T cells. Hair follicle itself also exhibits immune privilege, which is where an inhibitory tissue state is generated by the combination of low or absence of MHC class I, beta-2 microglobin, and MHC class II. There is also expression of no danger signal CD200 and local secretion of immunosuppressants such as TGF-beta 1, 2, alpha MSH, IGF-1, and cortisol. This specialized and actively maintained tissue environment promotes immune tolerance, inhibits inflammation, and restricts antigen presentation. In previous dandruff studies, the focus remained on dandruff scalp skin. Whilst this is important, the role of hair follicle also deserves to be thoroughly investigated in dandruff pathogenesis. Therefore, our working hypothesis includes the role of hair follicle in dandruff pathogenesis. We hypothesize that the normal scalp could enter a priming stage, which represents the non-lesional and peri-lesional skin of dandruff scalp. In this stage, there is an inflammatory milieu along with epidermal changes and epidermal barrier dysfunction along with microbial dysbiosis. Our main dandruff lesional part of the working hypothesis incorporates all the interlinking factors involved in dandruff pathogenesis, as well as the possible role of the hair follicle. First of all, the disruption within the epidermal barrier leads to the overcolonization of Malassezia species, both within the epidermis and within the hair follicle infobidulum, leading to bacterial species imbalance with higher abundance of Staphylococcus epidermidis and reduced abundance of Cutibacterium acnes. The breakdown of the host sebum lipids by Malassezia species and bacterial species results in the release of pro-inflammatory unsaturated fatty acids as the byproduct. This penetrates through the epidermal barrier and leads to epidermal hyperproliferation as well as further weakening of the epidermal barrier. It also leads to the activation of the immune response, resulting in inflammation. In dandruff epidermis, there is an increase of neutrophil chemoattractant interleukin-8. While it has been shown that the overcolonization of Malassezia species stimulates hair follicle keratinocytes to produce interleukin-8 and tumor necrosis factor alpha, this is currently unknown in dandruff hair follicle keratinocytes. Hair follicle keratinocyte may detect the changes in dandruff scalp microbiota, especially within the infobidulum, via toll-like receptor ligands, double-stranded RNA, bacterial metabolites, and cell surface antigen from either yeast or bacterial cells. This may result in recruitment of leukocytes and neutrophils via increased release of cytokines, chemokines, and antimicrobial peptide in dandruff scalp. The immune response could further promote epidermal proliferation and abnormal differentiation. The initial triggers of endothelial cell activation, leukocyte extravasation, and changes occurring within the sebaceous gland that alter the amount or composition of sebum and antimicrobial peptides within the dandruff scalp are currently unknown. Also, it is unknown whether or not the insufficient secretion of immune-privileged guardians such as alpha-MSH, TGF-beta-1, IGF-1, cortisol, and interleukin-10 by the hair follicle predisposes individual to dandruff development. To conclude, this novel working hypothesis shifts the focus to the hair follicle in dandruff pathogenesis. If confirmed, this provides new therapeutic targets for dandruff sufferers in the future.